Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we begin Chapter 12, Recent Innovations in Biotechnology. This chapter brings together environmental applications, advanced plant systems, regenerative medicine, nanobiotechnology, and synthetic biology. The central challenge is not to memorize a list of fashionable technologies. It is to understand what biological mechanism each innovation uses, what evidence supports its claim, whether laboratory performance survives scale-up, and how safety, ethics, accessibility, and environmental impact affect responsible use. By the end of the lesson, you will be able to evaluate biofuels through a life cycle, trace a crop trait from laboratory to field, explain how stem cells and biomaterials support tissue repair, connect nanoparticle properties to both function and risk, describe the synthetic biology build-test-learn cycle, and write balanced CBSE answers that distinguish demonstrated capability from speculation. Our roadmap begins with a common evidence framework for judging any new biotechnology. We then examine environmental biotechnology as a system for waste treatment, pollution control, resource recovery, and monitoring. Biofuels show why feedstock, pretreatment, conversion, co-products, and life cycle emissions must be considered together. Improved degraders and engineered pathways extend bioremediation, while plant biotechnology combines tissue culture, molecular markers, transformation, and genome editing. We next study regenerative medicine, stem cell-derived products, biomaterials, and tissue integration. Nanobiotechnology introduces size-dependent interactions used in delivery, imaging, diagnostics, and biosensing. Synthetic biology uses modular design and iterative build-test-learn cycles to construct biological functions. Finally, we follow an innovation from proof of concept through validation, manufacturing, and responsible deployment, always asking about controls, reproducibility, long-term safety, equity, and the limits of the available evidence. A new result can be scientifically exciting without being ready for clinical, agricultural, or environmental use. Proof of principle asks whether the proposed mechanism can work under controlled conditions. Validation asks whether independent measurements, suitable controls, and repeated experiments support the claim. Translation asks whether the effect survives larger scale, variable users, realistic environments, and manufacturing. Responsible deployment adds safety, cost, access, environmental impact, regulation, and public value. These stages cannot be collapsed into a single headline. A cell culture result does not prove patient benefit. A greenhouse trait does not guarantee field performance. A laboratory degrader may fail in heterogeneous soil, and a sensitive nanosensor may not be selective in a complex sample. Throughout this chapter, use an evidence readiness ladder: mechanism, control demonstration, reproducibility, scale, real-world performance, long-term safety, and social acceptability. Environmental biotechnology applies organisms, enzymes, and biological communities to wastewater treatment, pollution control, resource recovery, and environmental monitoring. A treatment must be described as a material flow, state the incoming waste, biological conversion, useful product, remaining effluent, sludge, emissions, and energy demand. Microbial communities can remove organic load, transform nutrients, or generate products such as biogas. Biosensors may detect a pollutant or biological response, but their sensitivity, selectivity, and field stability require validation. Resource recovery can improve sustainability when useful nutrients or energy are captured, yet every residual stream still needs safe management. Therefore, the correct question is not whether microbes are involved, but whether the complete system reduces environmental risk and resource use compared with alternatives. Strong evidence includes mass balance, field replication, energy and material accounting, residual toxicity, ecological impact, and stable operation over time. Biofuels are energy carriers produced from biomass or biological conversion. Examples include ethanol from fermentable sugars, biodiesel from lipids, and methane-rich biogas from anaerobic digestion. The conversion pathway alone cannot establish sustainability. Begin with the feedstock, how it was grown or collected, whether it competes with food, and how much land, water, fertilizer, and transport it requires. Pre-treatment may release sugars from complex biomass, but consumes chemicals and energy. Fermentation, or digestion has a biological yield and creates co-products or residues. Product recovery and distribution add further energy demand. A life cycle comparison therefore defines system boundaries and measures inputs, useful energy, emissions, land and water use, co-product credits and waste management. Second-generation feedstocks, such as agricultural residues, may reduce direct food competition, but collection, recalcitrance and seasonal supply remain constraints. Bio-based does not automatically mean carbon neutral or sustainable. Recent bioremediation innovation may use selected microbial consortia, improved enzymes, engineered pathways, OMICS-based monitoring or carriers that protect biological activity. Laboratory screening can identify an organism that transforms a pollutant, but the field contains mixed contaminants, changing temperature and pH, limited nutrients, competing organisms, and inaccessible sorbed material. A credible development pathway begins with parent compound loss and product identification in control cultures. Soil or water microcosms then test realistic matrices with sterile, untreated, and process controls. Pilot studies examine delivery, persistence, spatial heterogeneity, and containment. Field monitoring measures chemistry, biological activity, toxicity, and rebound after treatment stops. An engineered organism also requires assessment of survival, dispersal, and gene transfer. Innovation is valuable only when it improves the complete pathway to a safer endpoint, rather than producing a faster laboratory signal or transferring contamination into an unmeasured compartment. Plant biotechnology combines tissue culture, molecular markers, genetic transformation, and genome engineering to create or select useful traits. The evidence proceeds through levels. First, verify the intended molecular change and check likely unintended changes. Regenerate plants and confirm that the trait is expressed in the relevant tissue and developmental stage. Demonstrate stable inheritance across generations where inheritance is expected. Controlled growth studies test phenotype, fitness, and dose response. Greenhouse experiments add environmental variation, while replicated field trials examine yield, stability, pest or stress response, and interaction with local ecosystems. Food or feed use requires compositional and safety evaluation, and environmental release requires assessment of gene flow and non-target effects. A molecular marker identifies a sequence association, it does not by itself prove agronomic value. A strong answer traces the chain from DNA change to cellular effect, plant phenotype, field performance, safety, and stewardship. Regenerative medicine aims to restore damaged cells, tissues, or function. Its components may include therapeutic cells, a biomaterial scaffold, biochemical signals, and a controlled manufacturing process. A scaffold is not merely a support, its architecture, stiffness, porosity, and surface chemistry influence cell attachment, migration, and differentiation. Cells must have the correct identity, purity, and functional maturity. Signals must be delivered in an appropriate concentration, location, and time sequence. After implantation, the construct must integrate with host tissue, gain blood supply, communicate mechanically or electrically where required, and avoid destructive immune responses. Preclinical evidence therefore examines viability, phenotype, function, vascularization, integration, degradation of the material, and abnormal growth. A repaired appearance is insufficient if normal function is absent. Translation also requires reproducible manufacturing, sterility, storage, delivery, patient selection, and long-term follow-up. Stem cells combine self-renewal with the ability to generate differentiated descendants. Their potency differs, some can form many body cell types, while tissue-specific stem cells have a narrower range. In a therapeutic or disease model workflow, cells are expanded, guided through defined differentiation stages, purified and tested for identity and function. The final population must not contain an unacceptable number of undifferentiated cells, because uncontrolled proliferation can create tumor risk. Prolonged culture can introduce genomic or epigenetic changes, so stability must be monitored. Patient-derived induced pluripotent stem cells can support disease modeling and personalized testing, but a laboratory phenotype still requires clinical interpretation. Cell replacement additionally faces immune compatibility, delivery, survival, maturation, and integration. Release criteria should specify identity, purity, potency, sterility, and safety. Ethical questions vary with the cell source and intended use and should be addressed accurately rather than through broad claims. Nanobiotechnology uses structures at the nanoscale to interact with biomolecules, cells or tissues. At this scale, surface area becomes large relative to volume, and small changes in size, shape, charge or coating can strongly alter behavior. A nanoparticle designed for delivery may protect a cargo, improve solubility or carry targeting ligands. Yet after contact with biological fluid, proteins may adsorb to its surface and create a new biological identity. Particles may aggregate, distribute to unexpected organs, activate immunity or persist after the useful signal has ended. Characterization must therefore measure size distribution, shape, surface charge, coating, cargo loading, release rate, aggregation and stability in the actual medium. Biological evaluation follows uptake, intracellular location, function, clearance and toxicity across relevant doses and times. Small size is a design property, not evidence of safety. Nano-enabled applications include drug or gene delivery, imaging agents, diagnostic platforms and biosensors. Each claim demands different evidence. For delivery, compare free cargo, empty carrier and cargo-loaded carrier, measure uptake, release, biological effect and toxicity. A targeting ligand should be tested against a non-targeted particle and in cells with and without the target receptor. For imaging, establish signal strength, spatial accuracy, background, distribution and clearance. For biosensing, sensitivity describes how the signal changes with concentration, while selectivity asks whether similar molecules interfere. Calibration, blanks, positive controls and validation in real samples are essential. A low detection limit in buffer does not prove clinical usefulness in blood or environmental usefulness in turbid water. Manufacturing must also control batch-to-batch size and surface properties because these can change both performance and risk. Synthetic biology applies engineering ideas such as modular design, standardized components and modeling to construct or redesign biological systems. A simple biosensor circuit contains a sensor that responds to an input, a regulatory layer that processes the signal, and an actuator or reporter that produces an output. Parts cannot be judged in isolation. Promoter strength depends on host context, copy number, and growth conditions. Expression consumes cellular resources and can impose metabolic burden, slowing growth or selecting mutants that disable the circuit. Biological noise creates variation between cells. Interactions with native pathways may produce unexpected behavior. A complete design therefore specifies the host, input range, circuit architecture, output, response time, burden and failure modes. Mathematical models guide design, but experimental measurement must update the model. Predictability comes from repeated design and testing, not from assuming that cells behave like ideal electronic components. The build-test-learn cycle is the operational core of synthetic biology. During design, define the required input-output behavior and choose components using prior data or models. Build the DNA construct, verify its sequence and place it in a defined host. Testing measures dose response, dynamic range, background signal, response time, stability and biological cost. Controls include a host without the circuit, a circuit lacking the sensing element, positive controls and conditions that reveal unintended activation. Learning compares observation with prediction and identifies which assumption failed. The next design may change promoter strength, component order, copy number, degradation tags, or host background. Repeated cycles improve function and expose trade-offs. Containment should be designed in parallel through physical barriers, dependency on supplied nutrients, kill switches or genetic safeguards, then challenged experimentally. A safeguard that exists only on a diagram has not been validated. Many recent innovations arise from convergence. Genome sequencing identifies variants and candidate pathways. Transcriptomic, proteomic and metabolomic measurements describe biological states at different levels. Computational models can predict molecular structures, classify patterns, or prioritize experiments, while automation can build and test many designs. These tools expand observation and speed, but they do not replace experimental reasoning. A correlation in a dataset is not automatically a mechanism. Training data may be incomplete or biased, and a prediction outside the represented biological context may fail. High-throughput screens need appropriate controls, batch correction, and independent validation. A strong innovation links data to a testable hypothesis, perturbs the proposed mechanism, measures the expected consequence, and reproduces the result in a relevant system. Computation narrows possibilities. Biological experiments establish whether the predicted relationship operates in cells, organisms, or fields. Translation is a staged increase in evidence. Discovery identifies a mechanism or useful effect. Proof of concept demonstrates it in a controlled model. Independent replication and orthogonal measurements reduce the chance of artifact. A pilot tests the process at an intermediate scale and reveals mixing, delivery, contamination, variability, or supply problems. Scale-up requires quality specifications, reproducible manufacturing, and defined acceptance criteria. Field trials or clinical studies test the technology in the population and environment for which it is intended. Post-deployment monitoring detects rare or delayed effects. Failure at a later stage does not mean the early science was false. It may reveal a scale, context, manufacturing, or risk limitation. CBSE answers should therefore state the current evidence level and the next evidence required. Use words such as may, supports, or demonstrates precisely, and never present a future possibility as an established outcome. Responsible innovation asks not only whether a biotechnology can be built, but whether and how it should be used. Biosafety examines accidental harm to workers, users, communities, and ecosystems. Biosecurity considers deliberate misuse. Clinical applications require valid consent, privacy, and fair participant selection. Agricultural and environmental technologies require attention to gene flow, non-target effects, stewardship, and reversibility. Intellectual property can support investment, but may also affect affordability and access. Public engagement matters when communities experience the risks or consequences. Governance should be proportionate to the hazard and updated as evidence changes. Transparency includes reporting uncertainty, negative results, and conflicts of interest. Equity asks whether the technology addresses a real need and whether benefits are accessible. A balanced answer avoids both uncritical enthusiasm and automatic rejection. It identifies the benefit, evidence, uncertainty, safeguards, affected groups, and conditions for responsible use. For a CBSE answer, begin with an exact definition and biological scale. Explain the mechanism in causal order from input or structural property through cellular process to measurable output. For biofuels, draw the life cycle from feedstock to pretreatment, conversion, recovery, use, and residual streams. For a crop trait, trace molecular verification, expression, inheritance, greenhouse evaluation, field trials, and safety. For stem cell products, include identity, purity, potency, genomic stability, and tumor risk. For nanoparticles, connect size, shape, and surface chemistry to delivery or sensing, and include aggregation, distribution, and toxicity. Draw synthetic biology as sensor, regulator, and output inside a build, test, learn cycle with containment. Name the comparison controls and distinguish direct observation from inference. Conclude with readiness, reproducibility, scale-up, manufacturing, biosafety, ethics, equity, and a realistic limitation. Do not award novelty the status of proven benefit. Close your notes and rebuild the chapter. Environmental biotechnology treats waste, recovers resources, and monitors ecosystems, but complete material and energy flows determine benefit. Biofuels require life cycle assessment from feedstock through conversion and use. Improved bioremediation must move from culture to realistic metrics, pilot and field with products, toxicity and containment measured. Plant biotechnology connects molecular change to inheritance, phenotype, field performance, and safety. Regenerative medicine combines cells, biomaterials, and signals, while stem cell products require identity, purity, function, genomic stability, and long-term safety. Nanoparticle size, shape, and surface chemistry influence delivery, sensing, distribution, and risk. Synthetic biology combines sensors, regulation, and outputs through iterative build, test, learn cycles. Omics computation and automation accelerate discovery, but still require causal experiments. Every innovation travels from mechanism to reproducibility, scale, manufacturing, and real-world performance. Responsible use adds biosafety, ethics, equity, governance, and transparent uncertainty.